Lung cancer is one of the most devastating diseases of our time. Every year, millions of people around the world are diagnosed, and sadly, 2 million lose their lives to it. Despite decades of research, treatments remain far from ideal.
The first chemotherapy was hard with the cisplatin. It really took me down.
Treatments are often highly toxic, difficult for patients to endure, and in many cases, only partially effective. Even more, the process of developing new therapies is slow and still rooted in traditional trial-and-error experiments, which often generate results that are narrow in scope, not necessarily optimized, and cannot be generalized.
Lung cancer is not a simple enemy. It evolves, it hides, and it develops defense mechanisms that allow it to resist drugs and escape the immune system. For researchers, this makes lung cancer not just a disease, but a moving target.
This is the challenge we face. And just like in chess, overcoming it requires strategy, precision, and foresight. That is why we at Oncoligo believe a new approach is needed.
So I'm hoping to hang on long enough to get maybe an mRNA vaccine or something like that would really target the mutant.
The heart of our platform is the antisense oligonucleotide, or ASO. It's a short, synthetic, chemically modified strand of DNA that binds specifically to a gene's mRNA. Unlike most cancer therapies that act on membrane proteins and are limited to the few that are druggable, ASO works at the mRNA level, meaning virtually any gene can be targeted. Once bound, the ASO-RNA hybrid is recognized by RNase H, which cleaves the RNA while leaving the ASO intact. This allows the same ASO to be reused, fighting cancer from the inside.
But cancer genes mutate in many different ways. How can your ASO be precise and effective against all those variants?
That's exactly why we use the principle of synthetic lethality.
Synthetic lethality takes advantage of cancer-specific mutations. Take p53, for example, one of the most common tumor suppressors mutated in cancer. These mutations can take many different forms, but they all disable the same survival pathway. Cancer cells then become dependent on backup genes. By targeting one of these backups, for example RB1, we remove that safety net. Healthy cells, which still have functional p53, remain unaffected. In other words, one synthetic lethal partner could serve as a universal therapy for all p53 mutant cancers, something existing treatments can't achieve. That's the power of synthetic lethality.
Huh. But how do you plan to ever get your ASO near the cancer cells?
We conjugate our ASOs to an antibody.
Delivery is one of the biggest challenges in therapies. Our solution is an antibody that recognizes receptors highly expressed in lung cancer but not in healthy tissues, guiding ASOs straight to the tumor. Once bound, the complex is internalized by endocytosis, releasing the ASOs exactly where they are needed.
Even if you do all that, you won't kill all cancer cells. You need to recruit the immune system to really overcome it.
That's the antibody's hidden trick.
Built into the antibody's chain is a short peptide, a natural lung cancer-specific epitope that enhances immune recognition. When the antibody is internalized and degraded, this peptide is processed and presented on the cancer cell membrane through the MHC class I pathway, making the cell far more visible to cytotoxic T cells. As cancer cells die, dendritic cells capture and process the released epitope, presenting it via MHC class II to activate helper T cells. This amplifies and sustains the immune response, reinforcing the selective destruction of cancer cells. So this is how it all comes together. Once the platform enters the body, it travels to the tumor and is taken up by the cancer cell. Then the ASO is released and is free to silence its target mRNA. By exploiting synthetic lethality, this leads to cancer cell death. Meanwhile, the antibody is degraded into peptides. One of the peptides is the epitope, which is presented on MHC molecules, both on the cancer cell and through additional immune pathways, recruiting the immune system to attack the tumor. Our platform fights cancer on two fronts: from the inside by silencing essential genes and killing the cell directly, and from the outside by activating the immune system to recognize and eliminate any remaining cells.
But even if you manage to deliver your ASOs into the cancer cells, how can you be sure it will work precisely? Designing ASOs is not simple. You can't just guess the right sequence.
You're right, precision is everything, and this is where our model comes in.
We developed a model that predicts and optimizes ASO sequences before they are ever tested in the lab. We started with data from thousands of patent-reported assays containing detailed information on sequences, cell lines, and their properties versus inhibition. The data is high quality, yet biased, so results across experiments are not directly comparable. We modeled ASO inhibition with a simple differential equation. Its solution decays exponentially, and after correcting for concentration, we focus on the efficiency variable. To estimate efficiency, we asked, what makes an ASO effective? Over decades, knowledge came in fragments. We unified it into one predictive framework.
We grouped the features into categories. Hybridization-based features describe how strongly the ASO binds to its target mRNA. There is an optimal energy window, which we capture using nearest neighbor heuristics. For the top sequences, we run molecular dynamics simulations, modeling RNA helix changes and backbone flexibility, features missed by other energy models. Next, folding-based features reflect how loosely folded mRNA regions are easier to bind to. Off-target effects play a major role. Competing sites reduce potency and increase toxicity, so we detect them through a fast hybridization search that accounts for transcript abundance. Sequence-based features, RNase H1 and RNA binding protein motifs, along with base composition features like GC content and AT-skew, each contribute to ASO performance. Finally, we look at context-based features. Exons, introns, and UTRs each influence inhibition differently. And we also consider the target's own context, its mRNA abundance and half-life. By integrating all these dimensions, we can predict efficacy more accurately and design ASOs that outperform existing approaches.
To avoid overfitting, we use robust models and cross-cell line validation, ensuring predictions hold in new biological contexts. But the modeling doesn't end there. We connect the ASO module to other layers of our system. Through synthetic lethality network modeling, we map how genes interact and identify the most promising targets to silence.
And through antibody epitope modeling, we ensure that each ASO is delivered primarily to cancer cells and can also activate immune recognition. We optimized the antibody design with Mendel Bio and ESO tools to boost expression and identified optimal epitopes using the NeoEpitope platform from Tulor's lab. To pinpoint optimal epitope insertion sites, we used PSSM analysis. We then computationally estimated processing efficiency, presentation probability, and immunogenic potential, ranking epitopes by their likelihood to trigger a strong immune response. Together, these models create an end-to-end digital twin of our therapy. Our model doesn't just design sequences, it designs strategies. Each prediction brings us one move closer to beating cancer at its own game.
What makes our approach powerful is that it doesn't stop with one disease. The same principles we use to design precise ASOs can be applied to any RNA-based project, from therapeutic development to basic gene knockdown in model organisms. To make this possible, we developed a tool that's open to everyone, from iGEM teams to academic labs and biotech startups. With Tauso, anyone can design precise antisense oligonucleotides in just a few clicks. All the user needs to do is select an organism of interest and choose a target gene. The backend model will compute the most effective ASO sequences along with key predictive features. It's powered by the same validated model that guided our own experiments, trained on tens of thousands of experimentally tested ASOs, and proven to predict real knockdown performance. By sharing this tool, we aim to accelerate discovery, make ASO design accessible, and open the door for future therapies far beyond our own project.
Computational models can guide us, but in the end, we must test them in the lab. Our goal was to test every piece of our checkmate strategy. Our first step was to validate the core of Oncoligo, antisense oligonucleotides. Using our computational pipeline, we designed novel ASOs targeting MALAT1 and GFP RNA and tested them in lung cancer cells. The results were striking. Among the tested ASOs, some achieved stronger knockdown than the commercial control, validating our predictive model. In GFP-expressing cells, we also observed reduced fluorescence, confirming the ASO's effect at the protein level. Because off-target effects are a key challenge in ASO design, we also built a novel synthetic biology system to measure sequence competition between on- and off-target sites.
Next, we tested whether our ASOs could induce cell death, not just silence RNA. We targeted two genes that are synthetic lethal partners of MTAP, a gene often deleted in lung cancer. As an initial proof of concept, we treated MTAP-deficient cells with our ASOs and observed clear, sequence-specific cell death, captured here in real-time imaging. These results confirm that our ASOs act not only at the mRNA and protein levels, but also produce a visible effect at the cellular level, a key milestone toward demonstrating synthetic lethality in cancer.
We also wanted to explore whether ASO could serve as a gene silencing tool beyond mammalian systems, in simple organisms used for research. To test this, we engineered yeast to express humanized GFP, creating a cross-species platform. After several design, build, test, learn cycles, we achieved robust fluorescence, establishing yeast as a rapid, low-cost model for ASO testing.
After validating our ASOs, we focused on efficient delivery by optimizing Erbitux, an anti-EGFR antibody, for expression in CHO cells. Using the Mendel BioTool and the ESO system, we generated two codon-optimized variants. One showed nearly double the expression level of the original antibody, a major step towards scalable therapeutic production. Our next step is to conjugate the ASOs to the antibody and test its internalization, neoepitope presentation, and activation of immune cells, completing the path toward a full immune-driven checkmate against cancer.
But science alone cannot bring therapy to patients. That's why, alongside our research, we built a clear entrepreneurial strategy. We began by analyzing the unmet need. Many patients still lack effective treatment due to toxicity, resistance, and narrow coverage. Our market sizing revealed a $1 billion entry point, within a $17.7 billion global opportunity, representing our first product in lung cancer.
Our stepwise model combines early revenue from our ASO design engine with validation of our MTAP-targeted lung cancer therapy. Once proven, we can expand the same platform to other cancer types and even genetic diseases. SWOT and competitor analysis highlighted our key strengths: modularity, scalability, and precision, where traditional protein-based drugs fall short. We built a business and financial plan, performed market and risk analysis, and created a public pitch video to refine our roadmap. Our milestones take us from proof of concept to preclinical testing and partnerships that prepare us for the first clinical trials. As part of our strategic development, we filed a patent with Tel Aviv University, protecting our core innovation and marking a major step towards translating our concept into a real therapeutic platform.
To turn a plan into reality, you need the right people. Our team brings together engineers, biologists, mathematicians, and computational scientists, working side by side to merge innovation with impact and move Oncoligo closer to patients.
But we didn't do it all alone. We integrated insights from patients, clinicians, chemists, and biologists, refining our design and elevating our science. Alongside them, our board of distinguished professors provided ongoing strategic and scientific guidance. To connect innovation with real-world needs, we collaborated with Startup Nation Central, gaining valuable entrepreneurial insights. We also mentored and organized ISRAGEM, a local high school competition inspired by iGEM, introducing over 250 students from dozens of schools to synthetic biology. We shared our work at several conferences and even organized a mini international jamboree, bringing together 12 iGEM teams from around the world.
Together, all these experiences shaped Oncoligo and strengthened our vision for a future where cancer is no longer a threat.
Checkmate.
We are on a mission to cure cancer.